All right, Wickham. We're off to a great start. Welcome to our second to last lecture this summer. This one's on evolution. This one's one of my favorite. Uh, Evolution is one of these like touchy topics. There seems to be some sort of like atheistic versus religious split, and I think that's actually like a false dichotomy. You can still be very religious and believe in evolution, and it's not even a belief thing. Right? You can believe in God and you can still understand the facts of evolution. Evolution is like one of those things where it actually, I believe, to me, it invites more mystery and more spirituality around the presence of life on this planet because you realize you kind of get this glimpse into the resilience of each individual organism and their lineage as it's traced from now all the way back to like the first cell that was replicating. And it's like awe-inspiring to consider that every organism, living, breathing, moving, every living organism on this planet right now comes from a line, an unbroken chain of survivors over countless generations. And it's just like it gives you a little bit more of this like respect for what you see out there is you're like, man, like even though maybe this organism has only been alive for, a year or two, a couple months, it's like that organism is actually been alive for like 3.5 billion years and somehow managed to stay alive and reproduce. And that's pretty incredible. So, anyways, evolution is the change in allele frequencies in a population over generational time. And so we can graph it, but we can draw it mathematically and say, okay, well, delta is the change, so a change in allele frequency over generational time. So what might that look like? So if we had two alleles in our population, A and I keep saying A, big A and little a, or capital A, lowercase a, two alleles floating around in the population, we can measure their frequencies, and maybe they start out at 50-50. And then maybe the next generation, they're 45-55. And the next generation, maybe they're 49-51. And the next generation, they're back to 50-50. So the question is, well, did this population evolve with respect to this gene? Because we really have to look at it with respect to the alleles. And the answer is no, it didn't. I mean, yeah, there were some fluctuations in it, but the allele frequencies were not changing over time. Now, how about this one? Let's just say it's the same population, but a different gene. It's B gene. So they start out 50-50. Okay, 45-55. Okay, 41-59, 35-65. Did this population evolve with respect to this gene? And the answer is yes, this population did evolve. And so anytime that we see allele frequencies change in a population over generational time, as long as they're not reverting back to their original allele frequencies and they're not just fluctuating around an allele frequency, what I mean fluctuating around like a specific allele frequency, as long as you're not doing that, we can safely say like this population is evolving over time. It's changing. And so what that begs the question is it says, okay, well, what mechanism is driving these changes we see? Is this just randomness? Is this just like, oh, this individual happened to have four kids and, those four kids all have the capital A allele or the little a allele, which increases the proportional frequencies of that allele in the population. And this person just happened to have one kid. Um, and so is it due to randomness, like random in how much kids you have, randomness in who died? Like what is causing the change that we see? And so what we know is evolution is going to proceed by two mechanisms. The first one is natural selection, and we talked about that one just briefly, but genetic drift would be the other one. And genetic drift is exactly what it sounds like. It's just allele frequencies um, drifting away from what they were. So they are changing, but it's sort of like, you think like, okay, if you throw a piece of if you throw a piece of driftwood into the ocean, it just sort of floats around and kind of goes wherever the water does. There's nothing like, there's no motorboat on it pushing it a certain direction. And so allele frequencies can change on based on chance and just random fluctuations, and that's genetic drift. And then the other one is the actual mechanism where the environment is selecting for an allele that has an adaptive value to that population. Now, natural selection can proceed only by three mechanisms. Well, it can only work if these three mechanisms are in place. Uh, Variable heritable fitness, and we'll touch on what those are in just a second. Now, we can take the natural selection and the mechanisms it works under, and we can split it up and say, okay, there's actually three major pathways that selection can pressure a population or its alleles frequencies to change. One is directional, and that's where it's like pushing one allele frequency higher and then reducing the fitness of the other allele. The other one is stabilizing, and so that's one where it actually, the selective pressure keeps both alleles at a certain frequency in the population. And then the other one would be disruptive. So this would be like where the extreme phenotypes, the homozygous phenotypes, are favored, like they have a higher adaptive value. And the intermediates, or the hybrids between those two, have a lower fitness. And that would be something where you might start to see a population split over time and turn into two species. Anyway, so directional selection really has a lot to do with the adaptive value of a particular allele, and so, or the adaptive value of a particular set of behaviors or something like that. As long as there's a genetic component to it, it can be influenced by selection. And so we also see things like kin selection, where it's like, okay, the mother gives up, her fitness to save her kids. It looks like she's being altruistic, but since she's doing it to save her kids who share her genes, she's actually propagating and boosting their fitness. And so she's indirectly boosting her own fitness through her children or through her cousins or through her nephews and nieces. And so that's kin selection. And we see that a lot in the animal kingdom. I was just saying that like mom and kids, just as an example. And then sexual selection is another one. So that's where the mates are chosen. Like there's preferential mating. And usually it's based on some physical characteristic, which has a genetic component. And then we have stabilizing selection, which to me, that's just a heterozygous advantage. That's if you have both alleles, like if you have both copies of the allele, so you have the big A and the little a, you actually have a higher fitness than if you had just homozygosity on either end. And then disruptive selection, like I said before, can kind of lead to this splitting within a population in the same area, and that's called sympatric speciation. So what we're going to do is just dive in on each one of these little columns here. Genetic drift is the one we'll start with, and let's get there first. And we'll talk about bottleneck effects and founder effects and stuff like that as we talk about genetic drift. So what this slide is showing you is this is basically a layout for this lecture. It's basically saying, okay, this is how we're going to proceed and talk about each one of these things. So genetic drift is this idea, and I'm gonna talk about it over here, and then I'll let the slide go. But it's this idea. So up here in the right corner, I'm gonna get my pointer going. So up here in the right corner, we have a population of bunnies, and it's this case of it looks like complete dominance. So anytime you have a capital B allele, and look, they're calling it P here and Q here, like we did in the last lecture. So anytime that you have a big B allele, you're gonna have that brown fur color. And then the homozygous recessives will have the white fur. Well, let's just say randomly that only these 5 individuals breed. And it was based on randomness. It wasn't like, oh, these 5 individuals were like, I don't know, had the best mating dance and they got chosen. It was just that these other 5 individuals just didn't find a mate, and these 5 individuals happened to find a mate. So take these 5 individuals, these 2 homozygous dominants, and then these 3 heterozygotes, and breed them together. And so if you do that, then you'll end up with your allele frequencies changing. Notice they went from 50-50 down to 70-30. And then now let's say, okay, we make our new population. So some of the individuals that were breeding from this top population were heterozygous. So they made a white rabbit just based on, chance. But anyway, so they made one white rabbit, but now this next generation, the allele frequencies are a little bit skewed. Now our big B allele is a little bit more common, and it's not based on any adaptive value. It's just randomness of who got to mate that time around. Now let's say just by accident, these 2 rabbits, each with their homozygous dominant genotypes, they go on to mate. Well, what are all their offspring going to look like? Boom. And now you have an allele in this population that is fixed. So we have just the big B alleles out there, and the little b allele has gone away. And this wasn't based on selection. This was just who ended up happening, who happened to breed that time around. So genetic drift is a change in allele frequencies due entirely to chance. And this happens a lot. This is happening right now in humans. It happens in every population. The question is, how big is the effect of drift? Well, like we said before, the change in the allele frequency due to drift is not due to like the allele's adaptive value, the allele's ability to produce a phenotype that survives better than other phenotypes. It's simply by chance. But here's the kicker, right? So why are some populations affected more by drift than others? Well, if you only have 10 rabbits in your population and only half of those get to breed, just based on chance, well, then the chance that you select a perfect subset of proportional alleles to breed to the next generation is really small. It's sort of like if I told you, okay, open up a bag of M&Ms, right? And I say, okay, there's brown, yellow, red, blue, and green M&Ms in that bag. What I want you to do is just pull out 3 M&Ms. So you pull out 3. Well, you missed 2 of them. You know, maybe you pull out 3 red ones. Well, guess what? Now you're if those 3 red ones you pulled out are allowed to breed and make more M&Ms, you're only going to have red M&Ms. But if I said, okay, reach in this bag of M&Ms and pull out 50, the chances that you pull out a representative proportion of the alleles in that bag is going to be pretty high. So the bigger the population, the less genetic drift can really affect its allele frequencies. Which, if you consider this, right, go back to what we were talking about um, with Hardy-Weinberg equilibrium. Remember, one of the principles or one of the 5 rules of it was an infinite population size. Well, that's this opposite of this. If you have an infinite population size, if you happen to pull a few alleles out here and there, you know, and kill them off, it doesn't matter because you have an infinite population. But when you have a small population, like 5 people, and one of them gets hit by a truck, well, your allele frequencies change drastically versus if you had 10,000 people and one person got hit by a truck. Okay. Um, we see genetic drift a lot when we have colonization. And colonization is where you have a main group and then a smaller group, a subset of that main group, breaks away and goes and finds a new location. And so we're going to take a look at that here. All right, so founder effect. Oh yeah, sorry, I was moving on already. So we see it when a group splits off. We just talked about that. But then we also see it after like a major event, like a climate disaster or, you know, a tsunami or something, where it reduces the population size dramatically. So let's investigate each one of these real quick. So the founder effect is this idea here. And I actually did an image for this one because I feel like it serves, it's more telling than just words on a page. So let's say we have the mainland population, right? So this is where we have the bigger group. The allele frequency of our big A and the allele frequency of our little a are the same. They're 0.5, so they're equally represented in that population. But let's say that a small family breaks off. 3 small families break off. These 3 families, just if you were to reach into this population and say, okay, I'm going to pull out 5 alleles at random, the chances that you pull out 5 red alleles is not like super low. You could do it fairly easily. I mean, consider this, right? So 50-50 is the 50-50 is the proportion of each allele in this main population. That's sort of like flipping a coin. So you're like, okay, red equals heads, blue equals tails. Let's take 5 people out of this population. So that's like flipping a coin 5 times, and you just happen to get 5 heads. Same thing for this other group. This is like flipping a coin 5 times, you got 3 tails and 2 heads. Or flipping a coin 5 times and you get 5 tails. Each of these now represents a subset that's leaving, moving on, doing their own thing. So you have founding group 1 through 3. The frequency of the big A allele is fixed already in this one group. This other group is 60-40, and then the other one is the alleles are grouped. So when these groups go and find their new location they're going to settle, they're going to propagate the allele frequencies of the smaller group that left. So they're going to amplify what's there in the founding groups. And so this founding group 2 is going to have a 60-40. Founding group 1 is going to have all these red A alleles fixed, and then the other group will have all these blue A alleles fixed. And you might say, okay, so what does that mean? Where do we see this at? Well, the founder effect happened in the Amish. So the Amish are people that kind of follow a simple lifestyle. They prefer not to use electricity or automobiles and things like that. Um, and what's interesting about them too is that Amish rarely will breed with people outside of their own community. So what happened? Well, there was 200 individuals left a larger Amish population and moved out into Pennsylvania. Now, one of the leaders of the Amish community that moved out probably was polygamous. I'm just guessing. He might not have been, so don't quote me on that. But he ended up he was a carrier of a disease called Ellis Van Creveld syndrome. And what this is it creates polydactyly, which means multiple fingers. So there's jokes about like, oh, you're inbred, so you have like more fingers than, or more like fingers and toes. And that's, I mean, it's not necessarily true, but it comes from these sorts of things where you have the founder of a small community is a carrier for a particular type of disease, genetic disease, and then when that population starts to grow, well, his alleles end up becoming at a higher frequency. And so they start pairing up in the grandchildren. So you'll see a lot of this show up in the grandchildren and so on. And so today, the Amish in Pennsylvania have way more cases of Van Creveld than the mainland or the people outside of the Amish community, at least in this particular village in Pennsylvania. So let's talk about bottleneck effects. So a bottleneck effect is exactly like the image shows you on the right. The bottleneck effect is where you have, let's say, the original population is going to go through an event where its size is diminished drastically. So it's sort of, these are usually like a natural disaster, a tsunami, an earthquake, forest fire, things that can really hurt and take down a big population, but not based on their phenotypes. It's just like who happened to be in the wrong place at the wrong time ended up dying. And so what ends up happening is the people that are left behind, the survivors, are by not but by design, but by circumstance, they're going to have different allele frequencies than the original population. And so that surviving population is going to end up propagating the new allele frequencies into the future, and that could be good or bad. So, but what ends up happening is the gene pool is reduced in size, and that surviving population is going to have different starting allele frequencies. And what this does is it allows for some alleles that maybe would have been previously ignored in the original population. Well, now they're more present, and so they might gain traction. They might actually start to increase in frequency, even though they would have been probably removed by selection in the bigger population. And so what's interesting, and I want to point this out, is that mass extinctions, and we'll see this in our next PowerPoint, can often have this sort of bottleneck type effect on populations. All right, so we talked about genetic drift. We're going to move on to selection next. So natural selection is the second mechanism of evolution that changes allele frequencies. Now, there's 3 requirements for selection to operate. The first one is that in order for selection to favor one allele over another, is that the alleles have to produce different phenotypes. The alleles have to produce different trait forms. So an allele that produces a brown eye and an allele that produces a blue eye, those are 2 different trait forms caused by 2 different alleles. Now, if you had an allele that made blue eyes and then another sequence of nucleotides, like another allele that also made blue eyes, there's no way for selection to act on those differently because the phenotypes they produce were the same. So the alleles have to differ and the trait forms that they produce have to differ. So there has to be variety to select from. If I gave you a bowl of red M&Ms and I said, "Hey, pull out the best M&M," you're going to be like, "What are you talking about?" And that's what we mean by variable. And then heritable means, okay, if there is some adaptive value to the phenotype, if the phenotype performs better and survives better and forages better and mates better, that phenotype has to be transmittable to the offspring. It has to be something that can be inherited. So if somebody decides to dye their hair purple, and I'm just using a human example, but if somebody decides to dye their hair purple and that makes them super sexy and like they breed with a whole bunch of people, well, guess what? That purple hair phenotype dies with that person and it doesn't get transmitted to the offspring. So selections really cannot act on that phenotype. But if it's heritable, if it can be passed on in the genes, then selection can act on it. And then finally, there has to be differential fitness between the alleles and phenotypes they produce. Meaning, so for instance, if I have an allele that codes for brown eyes and I have an allele that codes for blue eyes, if brown eyes and blue eyes compete the same, then there's no way for one of them to be better than the other, or one of them to out reproduce the other, or one of them to out survive the other, just based on their characteristics. I mean, an individual is a collection and a composite of a whole bunch of alleles. So granted, selection is going to be acting on something, but for a particular allele and a particular trait form, there has to be a difference in the way that those phenotypes perform, both in how they survive, which could be running from predators, getting food, finding mates, or sorry, that would be reproduction, running from predators, getting food, finding shelter, finding water, that sort of stuff. And also those phenotypes would have to help them reproduce better, like find a mate and things like that. All right, so those are the three components of natural selection. But selection, like, so we talk about selection and sometimes we kind of lose sight of what it really is. We consider it to be this like mechanism that's, put in place by nature. But really, if you think about it, selection is just the environment that a phenotype is exposed to. So selection is the environment that selects. And the environment is everything you can think of that that individual is exposed to throughout their life. Climates, habitats, chemicals, predators, potential mates, food, shelter, relatives, strangers, everything in that individual. So everything extrinsic to the individual is their environment. So your environment isn't just like the desk that you're sitting at, the paper in front of you. It's also the people around you. It's your family. It's it's what's in your fridge, that sort of stuff. So the environment is that which selects, and it selects the best phenotypes. And I can't stress that enough. Natural selection doesn't directly affect the gene pool. It indirectly affects the gene pool. It acts on the physical expression of the alleles in the gene pool, and then it selects from those phenotypes that are produced the best. And then the reflectiveness of that is it's reflected in the change in allele frequencies. But selection can't act on the allele frequencies directly. It has to act on the phenotypes produced by the variety of alleles in a population. Okay, so the environment selects the best phenotype. All right, one type of selection that we talked about earlier was directional selection. And so that's the one that is going to select the trait form that has the most adaptive value. So the best trait form for that environment. And I can't stress that enough. So think about this. Humans living in Russia, in northern Russia, are exposed to a different environment than humans that are living at the equator. The selection of that environment is going to select different phenotypes because of the environment. So the environment's going to in the equator is going to favor maybe darker skin, more melanin in the skin to protect skin cells from cancer and things like that. Whereas like up in the Arctic, it may favor lighter skin, so you get more vitamin D content from the limited amount of sun you're getting. And so the environment is different for those two populations. And so the adaptive value is relative to the environment. So if I said, okay, this person has an allele that codes for dark skin, and you say, well, that's great. What's its adaptive value? Well, you don't know until you say what environments it's in. Is it in the Arctic or is it at the equator? And so that's what we're talking about. It's the best adaptive value for that particular environment. That's directional selection. So this type of selection favors or changes the frequency of one allele over the others. So let's get into that. All right, I'm going to pause this because my kid's yelling. Okay, he was telling me the cat was in the tree. So anyways, we talked about that. So when the environment selects the best trait forms for that particular environment and removes the worst, so there's the double-edged sword of selection. the trait forms that it favors are going to increase in frequency. And so we say they have adaptive value, and then the population as a whole adapts. But something to consider is that environments are always changing. So populations are always adapting. There's always a shift in selective pressure. And so directional selection, I threw up this image here. So one extreme trait is favored against another. So having a longer tail is favored over having a short tail. And so over time, the original population is this dotted line. And then the new population, because of, maybe something in the environment, like the snake, the predator in the environment. So the snake is the environment. The snake is the selective thing. Or maybe it's the birds, right? Because the wiggly tails look like snakes and scare birds away that eat lizards. And so the birds being in the environment are avoiding the longer tail because it looks like a snake. And so it's this idea that now the shorter tails will be attacked by birds and disappear. At the same time, the longer tails are favored and will survive and increase in frequency. So one of the things we talked about before was just this idea of kin selection and altruism. So one of the oddities of natural selection, of directional selection, are when behaviors like altruism are common in a species. And we see that in humans a lot, actually. There are, however, several reasons why there's this predisposition, that's what altruism is, to put one's own fitness at risk, and why that might have evolutionary benefits. So one of those reasons is that when the beneficiary of the altruistic act shares genes in common with the altruist. So if I'm saving your life and you and I share genes, and you have maybe a lot of kids and I don't have any, and I save your life, well, I am increasing my fitness indirectly because you are sharing your genes with me and I'm protecting my genes through you. So I'm look like I'm self-sacrificing and reducing my personal fitness, but it's actually benefiting me indirectly. And so natural selection would favor that behavior if the relatedness between the individuals was high enough and the fitness benefit to the people that are receiving the altruistic act. And so there we go. So it appears like altruism, but there's a payoff. And so we get into Hamilton's rule. And Hamilton's rule is this idea that if the relatedness between the altruist and the recipient is high enough, and the benefit, that's the fitness benefit to the recipient, is high enough, that it's greater than the fitness cost to the altruist, then altruism would be favored by selection. And that was Hamilton that wrote that up. And he wrote this up with social insects, because social insects are one of those things that are weird, because there's workers that appear to have given up their reproductive right. And when Darwin was writing his book on natural selection, he said, if anything is going to undo the work that I've done, it's going to be the ants, these social insects. Because at the time, he had no rational explanation for why natural selection would favor somebody developing that cannot reproduce. Like, if there was a gene that caused you to not reproduce, then that would end at you. Like, why would that propagate in a species? And so Darwin was confused by this, but Hamilton came along and gave us a mathematical reason why that might happen. And so basically, if the alleles for altruism are contained in the recipient, and the recipient's fitness benefits greater than the fitness of the altruist, then the altruistic favor is favored by selection, and will become common. All right. So the equation RB, let me go back just for a second. So the equation RB is greater than C is a mathematical explanation for why something like altruism might evolve. It says if the relatedness between the altruist and recipient is high enough and the fitness benefit is greater than the fitness cost to the altruist, then the altruistic behavior may evolve. So another oddity of natural selection is associated with the ability to choose a mate. It's associated with being able to oops. Okay. So this is where Members of one gender choose, and I mean that like sex, like is in males and females, like who's producing sperm and egg. So members of one gender choose which members of the other gender they can mate with. And so this is where certain trait forms are associated with reproductive success. And this is called sexual selection. So this is where mating stops being random. Okay, so it's going to take on a selective quality. What ends up happening is that members of the same sex, so if you are of the gender that needs to be chosen by the other gender, then you will compete within your gender to be chosen. So there's competition there as well. So what ends up happening, so typically the female in birds especially, but even in humans and some other primates, the female is the selective agent. In males, there's competition to prove their variation of the trait form is superior, and if the female chooses them, they will leave more copies of their allele for that trait form in the gene pool. So if the trait forms under selection trends towards more pronounced features, well, that can lead to sexual dimorphism, and that's where this comes from. So that's where the phenotypes of both genders are vastly different. And the way gender is assigned in the animal kingdom is by sex chromosomes. So what ends up happening is that some features in male birds are carried on the sex chromosomes and they're pronounced. And so you get this sexual dimorphism. And we see that in like Goldie's bird of paradise. The males have these giant red plumes and they're very pretty. And the females are less, I don't know what the word is, like flamboyant. So anyways, another example of this is where female peacocks preferentially mate with males who have more eyes on their feathers. And so the more eyes you have, the more likely it is you can breed. And if you have more eyes and that variant, so here we go back to selective. If you have an allele that codes for more eyes on your peacock feathers, and that allele is varied, meaning that there are other alleles that code for less eyes, and the female is choosing that, meaning that allele can get transmitted genetically to your offspring, and she's choosing that, that means already you have higher fitness because your environment, the female is the environment in this case. She's selecting the allele that's coding for more eyes, and then that allele is increasing in frequency for more eyes. And what ends up happening is that now you get this, it's possible you can get runaway selection. And that's where this exaggerated ornamentation by persistent directional female choice. So maybe that allele for more eyes goes to fixation because that's what she prefers. A mutation arises where maybe there's one or two more eyes on the plume, and then that one gets selected for. Maybe there's another mutation where it codes for even more eyes, and that one gets selected for. So it's this female that's chasing around these males with all these eyes on their feathers, and it looks ridiculous. We also see it in giraffes. So people used to think that giraffe necks, the long giraffe necks, was so they could reach the leaves on the top of the trees. In fact, I'm sure many of you also believe that as well. But what we found out is that the necks in giraffes is due to the males' competition for females. So the males will like whip each other with their necks, and whichever male is successful in whipping the other giraffe with his neck will get to mate. And so the giraffes with the bigger, thicker, longer necks get to breed. And you see what happens when that happens. All right. So another type of selection is stabilizing selection. So this is where heterozygous individuals are going to have a fitness advantage. Because the heterozygous individuals have both alleles, the frequencies of the alleles tend to stabilize in frequency. So intermediate phenotype has higher fitness. The extremes have lower fitness. The selection is going to stabilize the presence of both alleles in the population. Now, heterozygous advantage is where the heterozygous individuals have higher fitness than homozygous. There's also other aspects of heterozygous advantage. One of those aspects would be like it's not necessarily for fitness because the individual can't breed, but sometimes when you have adjacent species, like take horses and donkeys, for instance, they are so closely related that they can still produce offspring that can look, act, behave just fine. And their offspring they produce actually is more vigorous, meaning the mule can do more than a horse or a donkey by themselves. So the mule is like, it has the best features of both organisms. Now, horses and donkeys have a different number of chromosomes. So when the mules go to reproduce, they can't make a viable sperm and egg because when they go through meiosis, their chromosomes don't line up because they have half from a horse and half their other set of chromosomes come from a donkey, and they don't line up during meiosis, so they can't produce viable sperm and egg. But they do have this hybrid vigor, is what they call it. And so there is some advantages to being heterozygous. An example would be like with sickle cell anemia. You might say, well, cystic fibrosis too. Why that? Well, cystic fibrosis, it's been hypothesized, so I'm not going to say it's true or not, but that it seems to have a higher cystic fibrosis is higher in frequency than you would expect in the European population. And a lot of people think, a lot of not people, just scientists think it could be something with the Black Plague, like people that were carriers of cystic fibrosis were able to survive the plague, and so their allele frequencies increased due to their heterozygous advantage. And we see it in sickle cell anemia in populations that are infected with malaria. So here's how that works. If you're in a population that has mosquitoes that are carrying the parasite that causes malaria, you're probably going to have a higher proportion of sickle cell alleles in your population. And here's how that works. So you got one gene with two alleles, and this is for hemoglobin in the red blood cell. We'll say the wild type is a capital A. That means it's dominant-ish. It's actually incomplete dominance, but meaning that its code is for a normally shaped hemoglobin. Now there's this variant of that gene. So there's another allele, and we'll call it S. You could also call it little a too, but we're going to call it S for sickle. And S is not a good allele. It codes for, there's a lot of things that go wrong in the process of making hemoglobin with the S allele, and it codes for sickle-shaped red blood cells. And I drew a couple of images down here. So if you're a homozygous recessive, you have these sickly shaped red blood cells that don't bind oxygen well. They get clogged up in your spleen. They block vessels, blood vessels and capillaries, and it's just not a pretty picture. But if you have two of the wild type alleles, the two of the capital A, most of you guys have two of the capital A alleles, you just have normal red blood cells. Somebody that is heterozygous, though, they're going to have slightly misshapen red blood cells, and we'll talk about why that's an advantage. So if you look here, we have fitness on the y-axis and then just a bar chart here on the x-axis. And so the fitness for normal red blood cells suffers. Why? Well, because they're susceptible to malaria. Normal red blood cells, these nice plump round red blood cells, can be infected by the parasite that causes malaria much easier. So if you're in an environment, there we go again, the environment is that which selects. So if you're in an environment with malaria and you have normal red blood cells, you're actually just going to not have as good a fitness. Conversely, on the other side of it, if you have sickle cell anemia, you can't bind O2. I mean, I don't need to explain why that's a lower fitness. But in the middle, if you have these slightly bent red blood cells and you live in an environment that has malaria, well, you can bind O2 just fine, but you're also resistant to the malaria parasite or the parasite that causes malaria. And so you have a higher fitness. Well, what does that do? Well, that stabilizes the two alleles, A and S, in the population. Because when these individuals go and breed, two of their offspring, well, out of four offspring, right? If we drew the Punnett square, one of their offspring will have normal red blood cells, two will be heterozygote carriers, and one of them will have sickle cell. And so these guys suffer low fitness, these guys suffer low fitness. The two offspring that are heterozygous probably do better, and then they reproduce, and the same thing happens. So it's stabilizing the presence of those two alleles in the population. Okay, disruptive selection. All right, so disruptive selection is a major type of selection where the extreme phenotypes have higher fitness than the hybrid individuals. So individuals with an extreme phenotype will have offspring with higher fitness when they selectively breed with individuals of the same phenotype. What this does is this leads to a reduction in gene flow between the two extremes. So the intermediate phenotypes are disfavored, they suffer fitness than the two extremes. And so if you have homozygosity at one extreme and homozygosity at the other extreme, if you were ever to crossbreed between those, you would produce a heterozygous offspring and they would have a low fitness. So you're better off breeding within your own extreme phenotypes. And this can eventually lead to sympatric speciation. We'll talk about that in a second. So extreme phenotypes have higher fitness. Extreme phenotypes are rewarded with a higher fitness for mating with the same extreme phenotype. Gene flow decreases, and we'll talk about gene flow, so don't worry about that. And this will possibly lead to sympatric speciation. So these individuals can all be living in the same geographic area, but they might split into new species because anytime they crossbreed or hybridize, the individuals they produce have lower fitness. So let's talk about species, sympatric speciation, right? Gene flow. Let's touch on those next. So speciation is a process in how we get new species, but first let's talk about what a species is. So the biological definition is the one I'm going to give you. A biological species is a group of potentially interbreeding organisms that can produce fertile, viable offspring. So you might have the same species in two different areas where they don't necessarily see each other, and they're still the same species because they can potentially interbreed. And anytime they do interbreed, they do produce fertile, viable offspring. So I gave you the example before of a horse and a donkey can make a mule, and the mule has hybrid vigor. However, because horses and donkeys have different numbers of chromosomes, the mule cannot do meiosis. It can't do crossing over between homologous chromosomes. And so horses and donkeys are not the same species. They cannot produce fertile, viable offspring. But if we look over at dogs, which are a common household pet, you have a poodle and a cocker spaniel. You can breed them together and you can get a cockapoo, and the cockapoo can also produce fertile, viable offspring. So we would say the poodle and cocker spaniel, although different varieties of the same species, are the same species. Let's talk a little bit about gene flow. So gene flow is the enemy to speciation. If you have two populations, two adjacent populations, gene flow is the transfer of alleles from one of those populations over to the other population. Usually this happens through a process called migration. So consider back to our Hardy-Weinberg equilibrium. Remember, a population cannot be in Hardy-Weinberg equilibrium if migration is happening, because this brown beetle here has a different set of alleles than these green beetles here, and he might bring those brown alleles over into their population, changing their allele frequencies. So they are not at Hardy-Weinberg equilibrium when migration is happening. Gene flow is going to homogenize, meaning you have heterozygosity here, but it's going to homogenize the gene pools of populations that are next to each other. All right, so gene flow is homogenizing populations. So anytime that a mutation shows up in one population, as long as there's gene flow, it's going to move to the other adjacent populations. So migration is the ally of gene flow. Migration is what helps genes to flow and alleles to flow back and forth from population and keeps them all the same species. Migration is the ally of gene flow, but the enemy of speciation. You cannot have speciation when you have gene flow. They work in opposition to each other. So how do we get gene flow to stop? How do we get migration to stop? Well, one way is you can take a geographic barrier and plop it down right in the middle of a population and split that population from being able to interact with the other half. So, or maybe a large physical object splits one population into two, and now you have two separate populations. Now consider maybe some mutations show up in one population and another population, and then fast forward thousands of generations. Well, once introduced, if the two populations can no longer produce fertile, viable offspring, then we would say that they're two different species. So you have an original population, it's split by a geographic barrier. Changes take place in both populations. I know it looks like only one is changing, but trust me, they're both changing due to genetic drift and selection. And then when they get reintroduced back to where that barrier gets removed, they're so different. They've accumulated enough differences that they can be considered two different species because they cannot produce fertile, viable offspring. An example of this we see with the northern spotted owl and the Mexican spotted owl. There's a big mountain range that comes down through California. So you see the northern spotted owl is in the temperate, like Pacific Northwest regions. And then you have the Mexican spotted owl here is more in these deserty regions in Mexico, Arizona, and New Mexico, and parts of Utah and Colorado. But there's this huge mountain range here, the Sierra Nevadas and the Cascade Mountains, that separate these two species from each other. That these two species were likely one species. They're very similar genetically at one point in time, but now they are two different species due to allopatric speciation. Now, sympatric speciation is where, due to disruptive selection, the population, so this is where the extreme phenotypes are favored. The extreme phenotypes are favored, and they choose and preferentially breed within their own extreme phenotypic group. And they're in the same geographic area. So if they're preferentially breeding within those same preferentially breeding within their same extreme phenotypic groups, then there's no gene flow happening between the two extremes, the two phenotypic extremes. An example of this would be there's a lake in Nicaragua where you have two populations of cichlids. I don't even know if I say that name right. I've never heard somebody say that name. I just assume that's how you say it. Cichlids. I don't know. I'm gonna call them cichlids. I'm probably butchering that though. I've always seen the name, but then I've never like had to say it out loud until now. And I'm like, wait, is that right? So cichlids. We'll call them cichlids. So there's a group with thin lips, and then there's a group with thick lips. And the thin lips preferentially breed with only thin-lipped cichlids, and the thick-lipped ones only preferentially breed with say that 10 times fast, thick-lipped cichlids. And so eventually these two populations will probably split and become separate species. We can look at the Pundamelia cichlids as another example of sympatric speciation taking place right now. So this is an example of sympatric speciation taking place right now in the Pundamelia cichlids of East Africa. So it's another lake in East Africa. And the fish population is split into two groups. So there's the original Pundamelia, which lives up near the surface of the lake. And then there's this darker, dirtier orange variety called Pundamelia nyariri. The two populations were misidentified as different species and were named as such, right? That's why one is Pundamelia, the other one Pundamelia nyariri. And so they were misidentified. We know they were misidentified because of this. The researchers took the two types of fish. One was Pundamelia, the other one Pundamelia nyariri. And they took this fish and they split them into two tanks. So one tank got 5 Pundamelia, and 5 Pundamelia nyariri, and that was 10 fish in one tank. And then the other tank, they put the same thing. They put 5 Pundamelia, and 5 Pundamelia nyariri. Now the difference was on one of the tanks, they just left them in normal light. So that would be a control, right? They just, are these fish going to breed with each other in normal light? The other tank though, because one of the fish had orange on it, right, which is this nyariri, which lives closer to the bottom of the lake, they'd shown an orange light, a monochromatic orange light, which hid the orange colors of the nyariri. So the females of Pundamelia preferentially mated with only their own species in the normal light. So that's the control. So the females are choosing to breed within their own extreme, but in the monochromatic light where they couldn't see the differences in males, where the females couldn't see the differences in the males, they didn't have a way of preferentially mating, and the offspring produced were actually fertile. They could actually make offspring themselves. So these two species are actually two populations that are going under sympatric speciation in this lake in Africa. And so my question to you is kind of like, let's go back to the beginning. What type of scientific investigation did these two do? Did these scientists do? They had a control group, right? They tested one with an experiment. So that would be an experimental investigation. One had a control group or a placebo, and then the other one got the treatment. All right. So finally, we're kind of on our last slide here. All right. So once we get speciation, once speciation has happened, there's a couple of mechanisms that keep the two new species from ever blending back together. So one of those is there. You can basically look at it, what happens before the egg is fertilized and then what happens after the egg is fertilized. So one mechanism, which has many pathways, is what keeps the egg from being fertilized. And so that's called prezygotic. So zygote is when the egg is fertilized. So prezygotic barrier, they keep an egg from being fertilized. The other mechanism is after the egg is fertilized, what keeps the offspring from becoming fertile or viable. So we'll talk about that in just a second. But with the prezygotic barriers, so there's a couple different types. So temporal isolation is this idea that you have two species, maybe they were split allopatrically, there's some geographic barrier, and then one population's breeding times or flowering times started to shift to earlier in the year. So now when the two populations or two species are introduced back to each other, the one is flowering at a different time than the other, so it keeps the egg from fertilizing the sperm. That's temporal isolation. The mating times are off. Maybe one chooses to mate in the morning, one chooses to mate at the night, and so they never actually interbreed with each other. The other one is habitat isolation. So if the two species never are like have overlapping habitats, so they don't happen to bump into each other, well, then they'll stay isolated. A good example of habitat isolation would be the Pundamelia fish. One of them's living up in the upper shores of the lake, and the other one's living down on the dark, dirty bottom. So they don't really interact with each other, even though they're the same they're technically the same species, but called two different species, right? They're still isolated. They don't the extreme phenotypes are living in slightly different geographic areas, and they don't overlap. Another habitat isolation would be like if one organism is living in, like a squirrel is living in the trees, and then there's another squirrel that lives in holes in the ground, and they just, one is jumping from tree branch to tree branch, the other one's running around the ground looking for nuts that fell on the ground, and they don't ever interact. So that's habitat isolation. So they would never breed. The egg would never get fertilized. Another type would be behavioral. So maybe the mating rituals are slightly different. So one bird's mating dance just doesn't quite do it for his cousin, and so they don't breed, and so the species don't blend. Behavioral isolation could be lots of different things. It could be what time of, well, temporal would be what time of day. Behavioral would be like, I need to build a beautiful nest to present to the female so that she selects me to mate with. Well, this beautiful nest I built just doesn't do it for the sister species females. They just don't think my nest is cool. They prefer nests that are burrowed into a ground or up in a tree or something. And so that would be behavioral isolation. And then also finally, anything that keeps the egg from being fertilized could be if you have a sperm and an egg. Well, a lot of species, the zonapalooza on the egg can only be penetrated by the sperm, the head of the sperm of the same species. Like there's a compatibility thing there that allows the sperm to put its chromosomes into the egg. And so there could be a barrier there where the sperm just cannot penetrate the egg. Even if the two species breed together and they like have sex, then there could still be a possibility that the sperm does not penetrate the egg. Now looking over at postzygotic, so after the egg is fertilized, then the egg is going to start to go through replication. So it'll duplicate its chromosomes. It'll go through mitosis. Well, once those two chromosomes line up, it could be that the signals for development are all askew. Maybe their mutations between the two species have accumulated so much that the two sets of chromosomes can't communicate, or the transcription factors are not aligned just right to switch on the genes. And so although the egg does get fertilized, it doesn't go through complete development. It doesn't even develop into a viable offspring. Another possibility is that it's something like a mule where, yeah, it develops just fine, no problem. But when it goes to breed, it can't make a viable sperm or egg. And so those would be post-zygotic barriers. And that's sort of what creates species. That's this idea of, okay, we have selective mechanisms in genetic drift will move allele frequencies around in populations. Populations can either start to diverge based on their environments within the same geographic location, or they can be split and start to accumulate mutations in two separate geographic locations. And either way, those mutations accumulating, the species changing their behaviors, the populations changing their behaviors, all of these things add up to new species forming over generational time. And so that's kind of the essence of what selection and evolution is about. And when we get into our next lecture, this is like one of my favorites. When we get into the next lecture, what we'll do is we'll kind of take a look at what happened, like what actually transpired on our planet due to all of these principles that we just talked about in this lecture. So I hope you enjoyed it. I'll see you guys for the next one. Bye-bye.